Hello, good afternoon. Thank you for attending today's webinar, Integrating iPSC-derived functional models with immunogenicity assessment to de-risk antibody and bioconjugate candidates earlier, presented by Ncardia and Abzena. I am Salome and I'll be your host for today. So before we begin, I would like to share just a few housekeeping instructions. This webinar is going to be recorded and will be available on demand within 24 hours. If you have any questions, of course, please feel free to share it in the chat or via email after the session. All the questions are going to be answered in an email after the webinar. And now I have the pleasure of introducing our speakers. Joining us today is Sanne Holt, Associate Director, Immuno-oncology at Ncardia, and Erika Kovacs, Senior Director of Biotherapeutic at Abzena. And yeah, this is it. We're ready to start. Erika, over to you.
Okay, so thank you very much, Salome, and welcome everyone. The agenda of today's webinar will be, we will first review the FDA Modernization Act and the rise of NAMs. After that, I will give a very brief introduction to Abzena and our approach to early safety assessment strategies. Then I will move on to present some of our approaches we take for immunogenicity, immunotoxicity, and hematological toxicity de-risking. And at that point, I will hand over to my colleague, Sanne Holt from Ncardia, who will discuss some human iPSC-derived 7-tissue in vitro safety platforms and how you can use this to de-risk your program. So now I will start by discussing the impact of the FDA Modernization Act and the use of NAMs. So if you're attending this webinar, I'm sure you are very familiar that the FDA and some other regulatory agencies as well are really pushing to phase out routine animal testing. And this is not only because of ethical reasons, but also because these models are simply not very human relevant and they don't really predict clinical outcomes as much as we would like them to. So the alternatives that they suggest and that we are really working on providing for our customers is using advanced in silico tools. For example, Abzena has a tool for immunogenicity and developability assessments, and accompanying these is human-based in vitro models. These are typically more complex models, so 3D co-cultures, organoids, organs on chip type of assessments. And the final goal would be to provide a human relevant safety, immunogenicity, and PK assessment platform that is much more predictive than our current animal models. And I'm sure we can all agree that if we are able to do this, then there are multiple benefits here. Obviously, the quicker timelines, cheaper solutions, reducing the need for animal testing and the ethical aspects around that. And overall, this would provide a better decision-making platform early on to increase chance of success in the clinic. So biologics, the other aspect that we have to consider is that biologics are coming more and more complex. And that means that having an assay that is tailored for your particular MOA and modality is really key. And NAMs are very flexible, and in this sense can be tailored quite well to these very particular mode of actions. And this is what we have seen before, and we have developed, for example, kinetic assays versus endpoint assessments, model different cellular environments, for example, solid heterogeneous tumors, presence of absence of immune cells, and we have built quite a compelling case study in terms of Kadcyla and HER2 that actually shows that these type of NAM approaches can predict clinical outcomes that we later on see in the clinic. But overall, I think we all agree that drug development is a balancing act. So you want to develop a molecule that has high efficacy, stability, and really good safety parameters. But you need to balance that with the cost, time, and the feasibility of your development program. And this is where we truly believe that NAMs have a key role to play, because in the long run, once we really figure out how we can utilize these NAMs and link them to clinical outcomes, they will be invaluable in terms of providing this missing link sort of between the two. So now I will move on to briefly showcase Abzena and how what is our approach to early safety de-risking. Abzena is an end-to-end CDMO CRO. We offer comprehensive support from all of the steps, from all the way to from the very beginning of discovery all the way to manufacture. So we have partners who we do antibody discovery programs with, establish POC data, and then where we really add a lot of value and have a lot of expertise is sort of the middle part, the lead optimization phase and the candidate selection phase. So we have experts who will really guide you through the right bispecific design, optimizing the sequence, also bioconjugate expertise. And then we have a full suite of analytical and bioassays to really interrogate these top lead candidates to assess them from a functional viewpoint, also manufacturability and safety assessments. And once we've really narrowed it down and selected that top lead candidate, then we have an excellent cell line that will take you really quickly into cGMP manufacture, and we support you all throughout this process. And all throughout this process, we really focus on giving a holistic view and focusing on developability throughout. So what does that mean? We make sure that our molecule answers two key questions. Can we make it? That is, can we assess? Can we make it in a stable, high quality form that scales, is cost effective, and within the time frame that we need for the program? And also, does it work? Does it work within the as the expected mode of action? And also, is it safe for the intended target population? So for this, as I mentioned, we have an extensive suite of analytical and bioassay testing platforms. So in terms of binding and function, we will always tailor the package based on your specific MOA and modality. And then we can also run extensive manufacturability assessments. And then last but not least, focus on safety, immunogenicity, and immunotoxicity. And that will be the key part of my talk from here on. So why is reducing immunogenicity and as early on as possible so important? So in some cases, unwanted immunogenicity has very minimal clinical consequences, but that's not the case the majority of the times. Most of the times it will have an impact on safety and efficacy. So in terms of safety, the risk of these ADAs cross-reacting with endogenous counterparts can cause some quite adverse events, whereas in case of efficacy, the altered PK properties will lead to a reduced drug exposure. And of course, both of these will have a commercial impact, and a lot of times they can halt development programs at late stages. And in fact, one of the most common causes of failure in biologic therapy is the development of these ADAs. So what can we do early on to assess this risk? And here in this schematic, it sort of showcases the biological process of ADA formation, and all of our assays that I will demonstrate later on today are focused on the early events of this process, so the uptake and processing via APCs, that is the antigen processing cells, and then presenting these to CD4 T cells. So most of our readouts are CD4 T cell activation and proliferation. And this in the human body, in terms of clinical relevance, these have been shown very well correlated with the activation of B cells that will produce these ADAs. So in terms of the regulatory guidelines, most of the large agencies, FDA, EMA, and the like, have some sort of guidance on what's the best way to approach these early preclinical immunogenicity assessments. And what they all agree on is that the animal models are not very predictive and relevant in these cases. So what they recommend is a combination of in silico and in vitro approaches. So practically the NAM approach that is now coming really into forefront in drug development. So this is a summary of the full suite of assays that Abzena has developed to assess immunogenicity and safety concerns early on. In terms of the top row, you see the whole molecule assessment approaches. This is our EpiScreen PBMC assay, which is our most basic immunogenicity testing assay. You add your drug candidate to a culture of PBMCs, and after various time points, you take a cell sample and you assess CD4 T cell activation and proliferation that may be indicative of later on ADA formation. The DCT cell assay is a version of this assay that is specifically designed to test molecules that have a direct impact on T cell function. And then the cytokine release assay, which is really, really important in case of immunomodulatory molecules, because cytokine release syndromes and other infusion-associated reactions are perhaps the most serious adverse effects that can happen in terms of immunotoxicity. And once we've ran all these assays and we have identified that there is a potential risk for immunogenicity, then what we can offer is methods to identify these hotspots, these areas that may be causing the problems, and we can also suggest ways to engineer them out. So these methods are typically our isotope AI in silico platform, as well as proteomics and peptide-based approaches. So underlying all of this is our real extensive cell bank, PBMC cell bank that we have in-house. So we are an HTA licensed facility with various ISO accreditations. We regularly process blood, both from fresh whole blood as well as leukopaks. And what the leukopaks provide is a really extensive, large quality controlled bank from a single donor. We typically have 400 to 500 individual vials. So those can be used for really large studies. And this cell bank is also regularly characterized and QC'd. So we have standard peptides, antigens that we regularly test them for. We also run high resolution haplotyping on them, and we make sure that the 50 donor cohort that we select for these assays is a good representation of the European, North American, as well as the world population. So to give a bit of a more detailed dive into our most common immunogenicity assessment platform, the EpiScreen 2.0 PBMC assay. It's called EpiScreen 2.0 because a few years ago we have completely relaunched this platform, and instead of a bulk single endpoint readout assay, we now have a flow cytometry based approach. So we practically add your drug candidate to PBMCs, and then at various time points we take samples of these PBMCs. We can zoom in on specific cell populations, so most likely the target population of interest will be the CD4 T lymphocytes, and we can look at activation markers such as CD25, OX40, as well as proliferation, which we do with EDU incorporation. And the real power of flow cytometry is that you get quite a lot of information out of this. You really understand how your cell population is behaving, and you obtain graphs such as shown here at the bottom left, where you have the marker response on the y-axis, the proliferation response on the x-axis, and also incorporated is the information based on the stimulation index. And then you will have, we will be able to place your molecules sort of in this 2D space and give you a bit more detailed interpretation of what your molecule is doing from an immunologic viewpoint. So these assays are run monthly, and they will, and we will provide an IND compatible Word report in the end. Now, the other assay that I mentioned a few slides back that is really important, especially for immunomodulatory molecules, is the cytokine screen assay. So in this case, we take whole blood and we culture it with samples or test samples at different concentrations. And after 24 hours, we take supernatants and assess that with Luminex. And here I will demonstrate a case study that we ran for safety screening of a panel of bispecifics. So I don't have the time to go into that today, but we have done extensive functional assessment on a panel of bispecifics. And we have narrowed down the panel to these top 4 performing candidates, so I, A, E, and G, and assess them for safety concerns using the cytokine screen assay. And as you can see, the cytokine release profile of these candidates are quite different. The ones at the bottom, E and G, produce a lot of cytokines and as such are a potential safety concern, whereas I and A are very similar to our low immunogenicity, low immunotoxicity control Erbitox. So if we were to guide our clients on which candidates are the best to proceed with, we would suggest I and A. Now, last but not least, the other aspect that we often assess when it comes to immunological and hematological toxicity is safety screening for ADCs. So ADCs are typically administered intravenously, so they do hang around quite a bit in the bloodstream. And as such, it is quite important to assess whether they have any longer-term effects on the various cell populations. And in this typical case study, we've assessed 3 biologics, Kadcyla, Trodelvy, and Mylotarg, and their impact on B cells, T cells, NK cells, and monocytes. And we have found that Trodelvy and Mylotarg have an impact particularly on B cells and monocytes, whereas Kadcyla's impact is much more reduced, as we would expect. So I hope I've demonstrated how we can use all these early de-risking NAM approaches in terms of immunogenicity and immunological toxicity. And now I will hand over to my colleague, Sanne, who will discuss some other approaches that we can use to assess toxicity in other tissues other than the blood and PBMCs using NAMs. Thank you very much.
Thank you very much, Erika, for a lovely overview. I'll now introduce you to Ncardia, the company, and also talk you through some human iPSC safety models that we've developed. So Ncardia is a CRO. It was started in 2009, and it started as an iPSC-based cell supply company. We expanded after that into discovery services, and in general, we have about 15 years of experience now with over 30 scientists. As mentioned, we are an iPSC-based company, and everything we do is built around one core belief, is that human relevant models can produce better and more translatable data. We have a subset of services, so we mainly work with human iPSCs, but we also are expanding to NHPs and K1 cells to offer better safety services and assays. We still supply cells. We do custom iPSC services. We also have discovery services where we use the iPSCs in drug discovery assays. And then we have an arm where we do extensive safety testing for the drugs, also often iPSC-based models. For today, I'll be focusing on the safety services and one specific aspect of the safety services to give you a clear overview and link to the ADC talk in the background and see how we can utilize this in your drug development. So at Ncardia, we aim to help you utilize human biology to de-risk drug development early and with greater confidence. So we work across the full discovery to preclinical continuum, but as mentioned today, I'll be focusing very much on the preclinical safety testing. We believe that iPSCs give you a very physiologically relevant human-specific readout that animal models often struggle to provide. Particularly for ADCs and biologics, species cross-reactivity is often very limited or absent, where human iPSCs can really fill that gap and answer your safety questions. We can range from either helping you streamline your candidate selection or identify tissue-level liabilities before you go into expensive in vivo studies, or helping you build a safety assay for your IND application study. We really try and tailor to whatever is needed for your drug development. So Erika very nicely walked you through Abzena's toolkit for immune-mediated risks for ADCs or antibody-derived drugs. These are fully essential assessments which cover the immune side of the safety picture. And here I want to talk about an aspect in addition to that, which is the tissue part. When an ADC or a biologic obviously enters the bloodstream, it also enters your organs. So it will go through your heart, your liver, your lung, where indeed a very different question arises is, does the ADC or the biologic also trigger a response against your healthy tissues? We built a platform, and we built that platform based on TCR T-cell therapies, where they've shown really strong IND success with purely in vitro safety testing, because specifically in this field, it's really hard to find translatable animal models. And we believe that we can learn from these lessons and learn from these successes and fill that gap and utilize this for ADCs and antibody space as well. Together, the two platforms offer a more complete de-risking picture, ranging all the way from immunogenicity and blood cell toxicity to organ and tissue level safety, all in vitro and all very human relevant. So when we think about safety, you have your off-target and your on-target off-tumor safety, or on-target safety. Just to set the basics, overall, when you have your designed reaction, your efficacy testing, your antibody binds your cancer or cell that you're targeting, because that could be more broadly than oncology, and then you have your working product. Ideally, we then see that there's no reactivity whatsoever against healthy tissues. Unfortunately, that's not always the case. We know that ADCs or drugs in general have liabilities beyond what we mean them to target. Then you get to the final picture is where you have your unwanted reactivity. So we try and mimic that response and see if we can predict any clinical liabilities for unwanted reactions. This can be on target, so you have expression of your target on healthy cells and there is actually a functional response. Or there is an off target, so there is a different receptor, possibly from the same family, that's expressed where we see binding in this tissue and it actually triggers a functional response. What we've seen in the past is that quite often the question around healthy tissue liabilities is often answered around in silico analysis or tissue array assays. So very much looking at either expression or binding, but we felt that very much that functional element of does the binding actually result in negative toxicity or negative results is one that was missing. So this is our platform. We selected 7 tissues, 7 iPSC-derived cells that we felt represented either very major organs that form a big toxicity risks, or organs that are often come up in terms of expression of antigens that are commonly used in antibody therapies. So we have the cardiomyocytes representing the heart, we have the hepatocytes for the liver, we have the spinal motor neurons representing the CNS. The endothelial cells are vascular endothelial cells representing your vascular system. The alveolar epithelial cells is your lung, and then we have the intestinal for your intestine, and then finally we finish off with melanocytes, being skin also a tissue where we see a lot of adverse effects, and we felt that needed to be in a safety tissue panel. We have a readout with the Excelligence MP, which is impedance-based, and on the next slide, I'll cover that in a bit more detail. So just very briefly to align for people that aren't aware of the impedance system, the impedance system is a system that has electrodes in the plate, and it measures basically changes in electrical current when you seeded cells across your platform. It means that it picks up changes in morphology, so not just cell death and healthy cells, but also proliferation or changes in morphology. If they round up more because they become unhappy, it really will pick up very subtle changes, and it measures this across real time. So it's fully label free, it is non-invasive, so you can use the cells for a different endpoint readout, and you can basically track this for as long as you feel is necessary over time to give an accurate readout and pick up any changes in morphology of the cells. So in order to test our platform with the 7 tissues in an impedance-based readout, we built a case study. In this case study, we used cetuximab linked to MMAE, so a toxic payload. Cetuximab is anti-EGFR, and we started by selecting this antibody or ADC in this case. We selected this very deliberately as it's a well-characterized molecule, and we wanted to verify the platform against a known clinical adverse event profile. So we wanted to see if we could pick up what was reported on this drug and using real-world data. We had a look at in silico analysis, so had a look at where EGFR is expressed in the body, what tissues are relevant. And what we find is that the majority of the tissues in our panel are either, again, major organs that you want to de-risk anyway, or have expression of EGFR. But this in silico analysis can also be used to identify tissues in addition to this panel. This panel is not set in stone, so if there's a drug that has target expression in a different tissue, then it's very possible to expand this panel with other tissues that you feel are relevant for your drug. We had a look at target expression by ICC. So we placed the cells, we looked at expression of the target, one to then be able to link it to the functional readout, but also to be able to see that the cells are indeed representing what is also reported in literature in terms of target expression. We ran then the ADC on the impedance, and we did this for 72 hours in a CRC with all the relevant controls. So this is the data that you then get. I picked an example of a tissue where we actually saw reactivity of the ADC and nice dose response activity in that case. And we picked a tissue where there was no response at all. So the top graph is the epithelial lung alveolar cells, and we see the dark purple line is your doxorubicin, is your tox control, which is very highly toxic very early on. And then the lines underneath are the cetuximab, and you can see that there's a very nice dose response, not only in max killing, but also in onset of the response. So the lower doses take way longer before they start to show any reactivity. The lower graph is your vesicular endothelial cells, and actually there we see that indeed they are killed by the doxorubicin, but there's no reactivity whatsoever of the ADC or the control antibody. In addition to this overtime measurement, you can also pick a single time point. In this case, we picked 72 hours, and you can look specifically at 72 hours. What is my response within the different doses? And it just visualizes that dose response curve of the ADC and the killing really nicely, and also shows that at the lower doses, so 1 microgram per ml and lower, you don't actually see a response anymore. So I think that's important actually for this slide, is that we went very high in dosing. So the highest dose was 100 microgram per ml, which is not really clinically relevant, and it's a super therapeutic dosing. But we wanted to show that if we overdose, we can pick up certain effects because of expression of the target. And we then titrated it down to more clinically relevant levels. So if we make a heat map of the different doses across and then the tissues underneath one another, we actually see at the highest dose on the left, we see that for the alveolar, we see a clear toxic response at the highest doses. We also see that the hepatocytes triggered about 30% killing at the highest dose, and the melanocytes even had around 60% killing at that dose. The other tissues came back as no reactivity, which actually linked very nicely to the EGFR expression. If we then dilute down and we go to the right side of the heat map, we actually see that for all the different cell types, the tox disappears, except for the melanocytes, where we still, at the lowest dose, remain about 10% toxicity. So if we link that to the clinic, how do our results and our platform with this molecule relate to what's reported? And what we see is that skin, where we had our primary signal and high expression of EGFR combined with high toxicity at low doses, we see that actually over 80% of patients that received cetuximab also developed skin-related adverse events. For the lung and the liver, we saw a very clear dose-dependent response, where the high doses showed tox, but we lost that tox as we went to the lower, more clinically relevant dosing. And also there in the clinic, a similar thing was reported, where they saw that it was very clear dose-dependent responses within these tissues, and approved dose ranges were based on lung and liver reactions. We saw no activity in the heart, CNS, or the vascular systems, and also no adverse events in any of these tissues were reported in the clinic. For intestinal cells, we saw actually no response, so there was no direct target-driven response, where in the clinic there were some gastrointestinal events reported, but when diving deeper into the details of it, these were all immune-mediated. So none of these were direct epithelial injury due to the drug, which again matches with what we observed. So overall, this platform, we feel, fits the NUM space really nicely. Every model has its limitations, but we know for animal models, there is very big limitations. So here we feel this platform, because it's iPSC-derived, offers very human-relevant data. It is functional toxicity, so not just binding, and it gives a very tissue-specific insight into where potential risks are. You can always then follow up these risks with other assays, but at least it will give you put you in the right direction for where potential risks could be. You have multiple tissues in a single study, and they're very key relevant tissues. And I said the panel can be expanded with whatever tissues are relevant for the drug that you're testing. We also offer an IND-oriented output, so we try and make we make a word reporting that can then support your IND application to use that data directly in that. So overall, what have you learned today? Hopefully you have picked up that early de-risking approaches using NAMs can really reduce your timelines, your cost, and increase the chances of success in clinic. The human PBC-based in vitro approaches are key to assessing immunogenicity and also other immunological risks. The 7 human relevant tissue platform in a functional readout distinguishes from the current in silico binding solutions that are currently available. And iPSC tissues really reduce the donor variability and avoid long delays in sourcing primary cells, and also add a real aspect of human physiologically relevant results. We feel that overall these solutions are very good for alternative biologics with poor translatability to in vivo, and also fitting the NUM guidelines that are currently being set by a lot of the regulators. We want to thank you for attending. We hope you enjoyed the webinar. And as mentioned by Salome at the very beginning, if there's any questions for either Erika or myself, do reach out. The webinar will be available, and we're happy to have any discussions around any of the assays presented today. Thank you.